Hello everyone, welcome to GNANA DEEKSHA concept-based digital learning. I am Dr. D. Vasavi, lecturer in microbiology, Telangana Social Welfare Residential Degree College for Women, Mahabub Nagar. Can you guess what the picture shown here depicts? Yes, it is a bacterium surrounded by many viruses. Viruses cause many infections in humans, plants and animals. Similarly, they also affect the bacteria. Of course, this is a natural phenomenon. The viruses which feed on the bacteria are called as bacteriophages. The term is made from two words, bacteria and phagene. In Greek, phagene means to eat. The word literally means bacteria eater. In today's class, we will learn about the structure of LAMBDA phage, its life cycle, replication, and its significance. Before going to the structure, let us know how it is classified. LAMBDA phage is a type of virus belonging to the realm Duplodnaviria. Duplodnaviria consists of the orders Parvovirales and Herpesvirales. It belongs to the kingdom Euengongvirae, phylum Neuroviricota, class Cardoviricitus, order Cardovirales. Cardovirales contains tailed bacteriophages. Carda in Latin means tail. Cardovirales come under group 1 viruses in Baltimore classification scheme as they have double-stranded DNA and with 18,000 to 5 lakh base pairs in length. The virus particles have a distinct shape where each virion has an icosahedral head that contains a viral genome and is attached to a flexible tail by a connector protein. Under cardovirales, LAMBDA phage is classified into the family Syphoviridae. There are currently 783 species in this family, divided among 265 genera and 13 subfamilies. The characteristic structural features of this family are a non-enveloped head and a non-contractile tail. The genus is named as LAMBDA virus. There are 4 species under this genus, and Escherichia virus lambda is the type species among them. The bacteriophage lambda was discovered and reported in 1951 by Esther Lederberg while she was working with the bacterium E. coli strain K12. She has found clear circular areas in agar plates with lawn culture of E. coli K12. These clear areas are called as plaques, which are produced due to lysis of the bacterial cells by the bacteriophage. We can also observe the plaques in the laboratory by inoculating the phage sample onto a lawn culture of the host bacterium by double layer technique after incubation for 24 hours. Coming to the structure of the bacteriophage lambda, the lambda phage has a binal symmetry consisting of a head, which is also known as capsid, a head-tail connector called as collar, a tail and tail fibers. The head is icosahedral in symmetry and non-enveloped, measuring 54 nanometers in diameter and contains a double-stranded DNA. Virus with a head having 12 vertices or corners, 20 facets or sides, and 30 edges is known as icosahedral. The lambda phage head is made up of 8 proteins and it has a triangulation number of T is equal to 7. The triangulation number has been proposed for viruses by Casper and Klug in 1962, which indicates that the facets of a virus contain smaller triangles of numbers 1, 3, 4, 7, etc. The tail of the lambda phage is flexible and non-contractile, measuring about 150 nanometers long and 8 to 12 nanometers in diameter. The tail terminates in a single tail fiber as per Kasjan's and King's classification of 1975. However, several studies have shown that lambda phage has more than one tail fiber, or the side tail fibers, or STFs. The tail fiber measures 25 nanometers. The tail and the tail fibers are comprised of 12 proteins. Here we can see how the lambda phage appears under the electron microscope. The lambda phage infects E. coli strain K12, which is a gram-negative rod-shaped bacterium. It is motile by peritrichous flagellae, means flagellae surrounding the complete cell, and is a facultative anaerobe belonging to the family Enterobacteriaceae. The bacterial colonies on the nutrient agar appear large, circular, low convex, greyish-white, moist, smooth and opaque. It can also grow on McConkey agar, and the colonies are pink in color because of its ability to utilize lactose. This bacterium is widely used in molecular biology studies. Now, let us discuss about the life cycle of lambda phage. So, generally, the bacteriophages follow any one of the two types of life cycles, the lytic cycle or the lysogenic cycle. In the lytic cycle, the bacteriophage multiplies inside the bacterium and finally kills the host, whereby the progeny-phage particles are released out. So, it is also called as virulent cycle, and the phages are called as virulent phages. Whereas, in lysogenic cycle, the phage DNA integrates with the bacterial chromosome with the help of the phage-encoded proteins called as integrases and multiplies along with the bacterial cell without causing any lysis of the bacterium as long as favorable conditions exist. Here, the phages are referred to as avirulent or temperate phages. Examples of the bacteriophages which undergo lytic cycle are T2 and T4. Examples of lysogenic phages are lambda and P1. Let us see the different steps in the lytic cycle. The first is adsorption, next penetration, followed by multiplication, then assembly or maturation, and then the lysis of the cell and release of the progeny virions. The first step, that is the adsorption, is the attachment of the phage to the receptor sites on the host bacterial cell. Specific strains of bacteriophages can only adsorb to specific strains of the host bacteria. So, this is called as viral specificity. Now, next is penetration. In the case of bacteriophages that adsorb onto the bacterial cell wall, a bacteriophage enzyme drills a hole in the bacterial cell wall, and the bacteriophage injects its genome into the bacterial cytoplasm. In the next stage, in the multiplication stage, the phage genome synthesizes early proteins that break down the host DNA, allowing the phage to take control of the cellular machinery. The phage then uses the host cell to synthesize the remaining proteins required to build new phage particles. After that, assembly or maturation occurs. That means the heads and tails are assembled separately. The new genetic material is packed into the head and new daughter phage particles are constructed. After assembly, the host cells gradually become weakened by the phage enzymes and eventually the cell bursts. This is called as lysis. The lysis of the bacterial cell releases an average of 100 to 200 new phage progeny into the surrounding environment. We will now see the different stages in the lysogenic cycle. As in case of lytic cycle, the first stage is adsorption, then penetration of the DNA into the host cell. Here, what happens is the phage DNA circularizes. This circularized phage DNA is called as a prophage. The third stage is lysogenization, where the prophage integrates with the bacterial chromosome and the host bacterium is said to be lysogenized. At this stage, the host bacterium is called as a lysogen and is conferred by additional properties of the bacteriophage. The fourth stage is multiplication of the phage DNA along with the bacterial chromosome as the bacterium divides. This occurs for some generations, but when unfavorable conditions arise, the phage shifts to lytic cycle. Lambda phage is known to infect Escherichia coli K12, which initiates with the binding of the J protein of the viral tail fibers to the maltose receptor LAMB on the bacterium. This interaction triggers opening of the tail tube and injection of its genetic material through the cell wall into the bacterial cell cytoplasm. The phage DNA enters into the bacterial cell, starting with the right end of the DNA. Once the lambda phage has injected its genome into the host cell, development may follow one of the two different pathways, that is either lysogenic or lytic. In the lysogeny, a repressor protein and an integrase enzyme are synthesized by the phage DNA using host enzymes. This repressor molecule turns off transcription of viral genes. Then the phage DNA is circularized and the prophage is site-specifically inserted into the host genome. The bacterium continues to grow and multiply and the phage genes replicate as part of the bacterial chromosome. Lysogeny is most often seen during inoculation in the nutrient-poor media or when the host cell is infected with numerous copies of the virus. During lysogeny, the inserted viral genome remains in a latent state. Here, only the viral protein and C1 repressor proteins are expressed in low levels. Repression is established to prevent synthesis of deleterious phage proteins. So, deleterious phage proteins may lead to lytic cycle. Here, you can see the DNA of the lambda phage. It is double-stranded in nature with a size of 48,502 base pairs. The molecular weight of this DNA is 31 into 10 power 6 Daltons. The ends of the DNA are single-stranded with a stretch of 12 unpaired bases. These are called as cohesive ends or cos sites. At the center of the lambda DNA, you can find the attachment site represented by ATT. This is the site by which lambda genome integrates with the host cell chromosome. The lambda DNA is linear when it is present inside the phage, but it becomes circular when it enters into the host bacterial cell. This circularization is possible with the cohesive or sticky ends of the lambda genome. The circular lambda genome is now called as a prophage. This is the organization of different genes in the lambda DNA. This is genome organization. There are cos sites at the ends. Then, from left to right, we'll see the genes coding for the synthesis of head proteins, tail proteins, attachment site, genes involved in recombination with bacterial chromosome, then genes needed for DNA replication of lambda phage, followed by genes coding lytic cycle. The genes needed for lytic cycle include NUTL and NUTR. which are used for the transcription of the phage mRNAs followed by translation to phage proteins. These are the N utilization sites situated to the right and left. NUTR for right, NUTL for left. The gene ZIS is needed for the excision of the prophage from the bacterial chromosome to shift from lysogeny to lytic cycle. The CRO gene protein blocks the right operator OR, which will inhibit the transcription of C1 repressor, which maintains the lysogeny. And the genes needed for lysogeny are INT and C1. So INT stands for codes for integrase protein. The lysogenic life cycle begins once the C1 protein reaches a high enough concentration to activate its promoters after a small number of infections. The C2 and the C3 gene proteins are also needed to enhance the synthesis of C1 protein to continue lysogeny. Now, let us see how the lambda phage integrates with E. coli chromosome. The attachment site ATTP on the phage is represented as POP prime, and the attachment site on the bacterial DNA ATTB is represented as BOB prime. The sequences of B, B prime, P and P prime are quite different, but the sequences O in both the attachment sites are homologous. This O region is called core sequence, which is rich in adenine and thymine nucleotides. For recombination to occur between phage genome and the bacterial genome, pairing of the homologous regions occur. For integration of lambda genome into the bacterial chromosome, the integrase enzyme of the phage lambda recognizes these homologous sequences on both the bacterium and the phage. The bacterial protein IHF, that is integration host factor, is also required for integration to occur. Both integrase and IHF bind to the phage attachment site to form intazome. This integrase acts as a type 1 topoisomerase. The function of this type 1 topoisomerase is nick, rotate and rejoin. That means it causes a strand break in one strand of the double helix, rotates one branch of broken strand about the continuous strand and then join the ends with other strand. For this, the easily melted A plus T core sequences in each attachment sites open, and then complementary strands from different attachment sites pair to form a 4-stranded segment. Now, pairs of single strand breaks in homologous regions are made. Then, the topoisomerase activity of the integrase enzyme causes each double-stranded region to rotate and join with the opposite strand. The corresponding strands from the two core sequences with overlapping joints will now separate. The same process is repeated on the other side of the base paired region. The exchange of the segments between the bacteriophage genome and the bacterial chromosome results in a recombinant molecule with altered attachment sites, BOP prime and POB prime on the left and right side respectively. That is, BOB prime plus POP prime gives rise to BOP prime and POB prime. These are the prophage attachment sites. The integration itself is a sequential exchange via a Holliday junction proposed by Holliday, the scientist. The Holliday model represents the recombination between double-stranded, DNA molecules. There is a speciality in the integration of the lambda phage genome into the E. coli chromosome. The type of recombination seen between these two is called as site-specific recombination. That means the lambda DNA is inserted into the bacterial genome only at a specific location. This is in contrast with generalized recombination maintained by other types of phages like P1. The lambda DNA is linearly inserted between the gall and bio loci of E. coli genome. This gall gene is for the utilization of galactose by the E. coli, and bio gene is for the biosynthesis of biotin by the bacterium. The lambda genome or the prophage continues to multiply along with the bacterial chromosome as long as the conditions are favorable for the bacterium. The lysogeny is solely maintained by the C1 protein of the phage, where low levels of C1 are synthesized continuously from the promoter PRM, that is, the maintenance promoter. An important distinction here is that between the two decisions, the phage has to decide between the two cycles, lysogeny and lysis on infection, and continuing lysogeny or lysis from a prophage. The latter is determined solely by the activation of REC-A protein of the host. That means if it wants to shift to lytic cycle, this depends on the activation of the REC-A protein of the host. A cell undergoing an SOS response will always be lysed, as no C1 protein will be allowed to build up. However, the initial lytic or lysogenic decision on the infection is also dependent on the C2 and C3 proteins. In the cells with sufficient nutrients, protease activity is high, which breaks down C2 and leads to the lytic cycle. In the cells with limited nutrients, protease activity is low, making C2 stable. This leads to lysogenic life cycle and C3 appears to stabilize C2. If C1 protein predominates, lysogeny occurs. And if CRO protein predominates, lytic cycle occurs. A lysogen is generally quite stable and can replicate indefinitely without releasing the phage. But if the lysogenic bacterium is to be damaged, the phage can initiate a lytic cycle. This is called as prophage induction. You can see the signals for prophage induction. These are the damage to the DNA by the ultraviolet light, low nutrient conditions or starvation, and toxic chemicals like mitomycin C. When exposed to such SOS signals, the REC-A protein of the bacterium is activated to become a protease. This REC-A now cleaves LEX-A repressor of the bacterium, and at the same time it can also lyse the C1 repressor of the bacteriophage, which is required for the establishment of lysogeny. Cleavage of C1 repressor leads to induction, where the early promoters PL and PR, promoter left and promoter right, they become available for the RNA polymerase. The gene ZIS is transcribed from the PL left promoter, producing a protein called as excisionase. Excisionase binds to the integrase, and integrase recognizes the attachment sites BOP prime and POP prime and causes cuts in the core sequence. This produces back the BOP prime and POP prime sites, reversing the integrase reaction. So these two are reversible reactions. The excision of prophage leads to lytic cycle. The bacterial chromosome is disintegrated. The synthesis of heads and tails of the phage occur inside the bacterial cell. The lambda DNA replicates initially by theta replication, which lasts for 5 to 15 minutes after entry into lytic phase. The replication starts at the replication origin, ORI, located within the gene O and proceeds bidirectionally, that means both sides, forming two replication forks. The replication intermediate appears like the Greek letter theta. Hence, this replication is called theta replication. The DNA dimers produced are resolved by the host topoisomerase 4. After 15 minutes of shifting to lytic cycle, the replication now shifts from theta mode to another mode, that is rolling circle mode or sigma replication. Here, the replicating molecule appears like the symbol sigma. This mode of replication produces concatenamers of the daughter DNA molecules. Means continuous synthesis of the daughter DNA molecules occur on a template strand where the daughter molecules are linked to each other. Then DNA maturation of the lambda phage occurs where the concatenamers are excised or cut to single genome length and then packaged into the empty capsids which are already synthesized. At least 11 proteins are required for the assembly of tail, which occurs from the tip protein. So it starts from tip to the gradually the proteins are built one on the other. The tubular part of the flexible tail is built from 32 discs. Finally, the tails are attached to the heads. The lambda phage produces proteins called as holins, which form a gateway for the lytic cycle. The height of a holin ring is 4 nanometers, which spans the lipid bilayer of the inner membrane of the host bacterium. This protein forms non-specific channels in the bacterial cell membrane. Through these channels, the host cytoplasm leaks out into the periplasmic space. The lambda glycoprotein R acts as a lysozyme, which can degrade the peptidoglycan layer of the bacterium. The combined action of holin and R protein causes lysis of the host cell and release of the progeny phages. The burst size of the phage is approximately 100 block-forming units per cell per generation. That is, burst size is the number of phages released per cell per generation. Now let us know the significance of the bacteriophage lambda. Phage lambda was in its golden age at the center of the molecular genetic studies in the late 1970s and early 1980s. It helped in understanding the molecular and genetic control of gene expression, that is production of the protein. Phage lambda DNA was one of the earliest model systems for studying the physical nature of DNA and genes. The lambda virion chromosome was also among the first natural linear DNAs whose end structure was understood in detail, the cos sites. Lambda was directly responsible for the discovery of the first transcription controlling factor Rho. Lambda phage is involved in transfer of genes from one bacterial cell to another by a process called as specialized transduction. Specialized transduction is that where the lambda phage integrates into a specific location, as we have seen in the site-specific recombination of the bacterium, and then transfers only specific genes from one bacterium to another bacterium. In this case, for example, the Gal gene or biogene can be transferred to another bacterium. Lambda phage was used as a model system to characterize eukaryotic double-stranded DNA viral assembly. It is also used as a cloning vector in recombinant DNA technology. There are two types of lambda vectors, the insertion vectors and the replacement vectors. The major advantage of the lambda phage vector is its high transformation efficiency. So it has a transformation efficiency of about 1000 times more than the plasmid vector. Let us have a quick recap of what we have learned today. The lambda bacteriophage is a linear double-stranded DNA virus with an icosahedral capsid and a flexible non-contractile tail. The phage infects the bacterium E. coli K12, which is a gram-negative bacterium belonging to the family Enterobacteriaceae. The phage attaches to the bacterium by its tail fibers and injects its DNA into the bacterial cell. Soon after this, it enters into lysogeny and multiplies along with the bacterial DNA for several generations. When there is a threat to the life of the bacterium, the phage shifts its life cycle from lysogeny to lytic cycle. The C1 protein plays a key role in taking the decision between the two cycles. The phage synthesizes its heads and tails separately. The DNA replication occurs both by theta and rolling circle models. After that, the DNA is packaged into the empty capsids and the tails are attached. The bacterial cell is lysed and the contents are leaked out. The progeny phages are released out with a burst size of approximately 100. The significance of lambda phage is it is used for molecular genetic studies and also as a vector in recombinant DNA technology.